and now I will start with what are cancer stem cells and what is my experience with cancer stem cells till date. Before sharing my experience, I will just like to highlight and define what are cancer stem cells for those who have no idea about this particular cell population. So first of all, I would like to share with you all that why we all should know about this particular subpopulation of cells, which we call as cancer stem cells. So we all are much familiar with cancer. The terminology cancer gives us a shock. It is really disheartening or really very shocking, very like whenever we hear about this term, we are never happy. Not even our, like, if one of our family members, friends, even distant relatives, and even if we see any case on social media, just a second. Even if you see any case on social media, we get much sad and shocked. So, first of all, why we all should know about cancer stem cells? We all know that cancer is a dreadful disease, and till date, we have no cure for this. So, most dangerous thing about this disease, cancer, is, cancer is cured, after the first cycle of chemotherapy or 2 cycles or 3 cycles, depending on the stage, depending on the case, it is cured easily. But what happens after a few months, after a few years, this dreadful disease comes back into life and then it finishes the patient. So that's why we should all become much more aware, much more knowledgeable about this particular cell population, which are cancer stem cells. So cancer stem cells is such a population of cells which is present in a tumor mass, which is responsible for causing cancer relapse. And relapse is a dangerous thing. Once a patient undergoes relapse, it is very hard to go back to the healthy phase of your life. Today morning only, my mother was sharing a story with me that, she was distributing report cards. She's a teacher now. So she was distributing report cards in her school, and like one of the students' mother, she was being detected with ovarian cancer few years ago, and she was totally fine after the chemotherapy. She was totally cured. But today only, when that particular student came to my mom to collect the report card, and he shared that he didn't come with his mother, unfortunately, this time. He was accompanied by his grandfather, if I'm not forgetting it. So her mother, even after complete cure with ovarian cancer, she was completely fine, doing well with her life. She was back in her normal life. But now she is being detected with the end stage, that is stage 4 of cancer. So this means that even no matter how much we try to like give therapy, no matter what are the therapies which are available in the market now, there is no proper cure for cancer till date. And the relapse, whenever a patient is undergoing relapse, it is very hard to like cure that particular patient. So this story in the morning makes my heart much more shattered. And I was thinking that today only I will be sharing my first webinar of life. So, and today in the morning only I get to know this story from my mom. And it was really heartbreaking for me. Like a girl who is only 30 to 32 years old is now suffering from last stage of ovarian cancer. And it is really very disheartening. So I would like to share that most of you are not familiar with this particular cell. So I will keep my description much more basic. So cancer stem cell, as I just said, it is a small population in the tumor mass, which is responsible for causing cancer recurrence. Cancer stem cells has the power to give rise to the entire heterogeneous tumor mass. So these cancer stem cells, you can see here, these purple cells, these are cancer stem cells. They can undergo 2 kinds of cell division. So one is they can give rise to themselves by symmetric cell division, which we also call as self-renewal. On the other hand, you can see that it can divide in asymmetric mode. That is, it will give rise to its own cell and it will give rise to a progenitor cell. Now, this progenitor cell will finally give rise to the mass of the tumor, that is the heterogeneous mass of the tumor, will be built with different types of cancer cells at different stages. So that is how a cancer stem cells undergo cell division. So why there is a tumor relapse even after the conventional therapy of radiation and chemotherapy? We can see that a tumor bulk once undergoes conventional anti-tumor therapy, the non-cancer stem cells. What are the non-cancer stem cells? The rest of the cells of the heterogeneous tumor mass, which are non-cancer stem cells, are basically the tumor cells. The entire bulk shrinks. The entire bulk is removed, is dissolved from the patient's body once a patient undergoes chemotherapy. Then why cancer comes back? Because a certain population of cells resides which are not detectable. They are not detected once a patient is cured with chemotherapy. They remain in a dormant stage. They maintain their dormancy in the presence of chemotherapy, and they are ultimately responsible for give rise to the tumor recurrence. As you can see here, all the other cells are dying in the presence of chemotherapy, but these orange colored cells, which we can call as cancer stem cells, they are not dying in the presence of chemotherapy, radiation therapy, or any other type of therapy which are available in the market till date. Just a second, I think somebody is joining.
Yeah, that's Pallavi.
Yeah. Okay, so you can like mute yourself. So as I was saying, that these orange colored cells, they are not affected in the presence of chemo or radiation. And they are called as cancer stem cells. They survive, but they are present in such a less number once a patient undergoes chemotherapy and is completely cured with cancer. These particular cells resides in a dormant stage. They maintain their quiescent nature, dormancy. And then these cells, depending on the time frame, like it may vary from months or after years, it will give rise to the entire heterogeneous tumor mass, not only in that particular place where the cancer originated first, but at the stage when cancer is again detected, undergoes relapse, when a patient undergoes relapse, it will be detected, the tumor mass will be detected in any part of the body. So thus, a patient undergoes cancer relapse, and which is really very disheartening. So what are the nature of these cancer stem cells? As I was sharing in my first slide, that how cancer stem cells divide. Either they will give rise to their own self, by undergoing self-renewal or symmetric division, or it will undergo asymmetric cell division. That is, it will give rise to its own self, one copy of itself, and the other copy will be of a lesser differentiation. Like it will give rise to progenitor with less stemness. This progenitor will further give rise to the cancer cells, differentiated cancer cells. So basically, cancer stem cells, they share so many properties similar to normal stem cells or adult stem cells, which are present in our body. But the problem with these cancer stem cells is that, unlike normal stem cells, maintains homeostasis, but these cancer stem cells do not maintain homeostasis. They are not able to maintain the balance between their own cell pool and the differentiated cell. That's why these cancer stem cells are extremely dangerous. So then the question arises, as I said, that these particular cells are present in much lesser amount in the entire heterogeneous tumor mass. If a tumor mass contains 100 cells, there will be 2 to 3 cancer stem cells within the entire heterogeneous tumor mass. But this number can vary depending on the stage of the cancer, grade of the cancer. This 2 to 3 number of cancer stem cells can rise to 50, 60, or up to 80 out of 100 cells in the tumor mass. So how to identify cancer stem cells? Those who are familiar with these cancer stem cells, they can easily say, like we can identify them with flow cytometry, Western blot analysis, ALDH assay, ABCG2 detection. But detecting cancer stem cells is really difficult. Till date, there are several research articles. They're not sure about what can be the exact marker set to detect cancer stem cells in a particular organ. Like cancer stem cell markers also vary depending on its origin. Like if it's breast cancer, the marker set is different. If it is cervical cancer, the marker set is different. And more markers are, coming. And since research is going on extensively on these particular cells, so every day we are getting a new information about this cancer stem cells. So nothing is defined yet. So we can just like guess, and based on the previous research articles, we can just say that yes, for breast cancer, we have this particular subset. For lung cancer, we have this particular subset, but more is to come. So here in this slide, you can see that I have shared what are the different types of CD. CD markers, cluster differentiation expressed. Just a second. Okay, so these are the markers to detect, you can see here also, here for colon cancer, head and neck cancer. So there are different subset of markers which are used to isolate different types of cancer stem cells from different specific organs. And then comes the major question, like why these cancer stem cells are not killed or not sensitized, not removed from the body, even in the presence of chemotherapy or radiation therapy. Why is the therapy resistance? So these are all the factors which confers chemoresistance to these cancer stem cells. One is their dormant nature, the tumor microenvironment, which we also know as vascular niche. Another one is they are even stable in the presence of hypoxia. As I say, cancer stem cells are very lesser in number in the entire tumor mass, and they reside within the core of the tumor. But even in the core of the tumor where there is no oxygen at all, these cancer stem cells can survive even where there is no oxygen. And that's how they are hypoxic stable. Also, upregulation of anti-apoptotic markers. Like this cancer stem cells upregulate all the anti-apoptotic markers. And also they have a special morphology and they have an increased condition of DNA damage repair pathways. Like all the DNA damage repair pathways are extensively increased here. Like they cannot undergo cell death so easily because the DDR mechanism is extremely high there. At the same time, the drug efflux pumps, which are ABCG2, MRP1, several pumps are present on the surface of these cancer stem cells, which throw away the chemotherapy drugs from inside these cells to outside, and thus they are resistant to chemotherapy drugs. So first, I will focus on what we know as vascular niche and dormancy in cancer stem cells and what is tumor microenvironment. You can see that in the tumor microenvironment, we have different types, different nature of cells. And a particular tumor microenvironment composes of a heterogeneous population, cells coming from different background. So, it's a teamwork. Basically, a tumor formation, progression, metastasis is a teamwork of all these cells which are present within this tumor microenvironment. You can see this like the light orange golden cells are cancer stem cells. They are supported by, accompanied by all other immune cells, which are not anti-tumor but pro-tumor. And this pro-tumor nature of all these immune cells is regulated by these cancer stem cells. They shed certain factors, they shed certain cytokines, certain chemokines, molecules via exosomes or directly to convert all the anti-tumor immune cells into pro-tumor in nature. So it is very difficult to manipulate them directly. So what is hypoxia? Inside the core of the tumor, I'm not talking about the age of the tumor. Inside the core of the tumor where oxygen concentration is very less. Cancer stem cells resides in that harsh condition of less oxygen availability. And thus, they are hypoxia resistant. They can survive even in the presence of hypoxia. The more is the hypoxia, the more cancer stem cells can survive. So there are certain factors. I will not go into much details of the science, like which factors are responsible to upregulate all the particular genes which helps in sustenance of this cancer stem cell population. And then how they escape cell death and how they escape apoptosis. It is not easy to kill these cancer stem cells, unlike cancer cells. It is very easy to target the tumor mass, but it is very difficult to target the cancer stem cells because they smartly and intelligently overexpress all the anti-apoptotic proteins. So we can say that yes, cancer stem cells are extremely and highly intelligent. Depending on the niche, depending on the microenvironment, depending on the nature of therapy they are getting, they can quickly manipulate themselves and protect themselves. So these are the morphology of cancer stem cells. Whenever we culture, like as a cancer biologist, whenever we culture cancer stem cells in laboratory condition, I'm not talking about how they look inside the body, but whenever we culture, just a second. Whenever we culture cancer stem cells in laboratory condition, they look like this. So these are the spheroids. Like the spheroids culture is a 3D culture of cancer stem cells. It is done to enrich the cancer stem cell population. And these are normal cancer cells. So this is how morphologically they look distinct in vitro. And also, I just shared that how they look in laboratory condition, but inside our body, the cancer stem cells, they have a large nucleus with a lesser cytoplasm. And most of the cancer stem cells are mesenchymal in nature because cancer stem cells are majorly and highly responsible for metastasis. So to perform metastasis, they had to change their morphology into mesenchymal nature so that they can migrate through the blood vessels and quickly go and invade all the other organs of the body by mesenchymal to epithelial, by epithelial to mesenchymal transition. And whenever they get the organ, they again undergo EMT, MET, that is mesenchymal to, I'm just getting confused about EMT and MET, but as you can understand, cancer stem cells had to change their morphology while migrating so that they can easily, quickly pass through the bloodstream, the blood vessels, and go and invade the new organs. So now comes, what is aldehyde dehydrogenase in cancer stem cells? So, aldehyde dehydrogenase is such an enzyme which converts retinal to retinoic acid inside the cells. And this retinoic acid is majorly responsible for stemness, survival, and growth. So cancer stem cells, one of the way to detect cancer stem cells is aldehyde dehydrogenase assay. I haven't described the assay here, but with aldehyde high and low cells, you can distinctly separate the cancer stem cells from the cancer cells. And the major reason behind drug efflux, which I just said, the ABC transporters. The ABC transporters, the multi-drug resistant proteins, are highly overexpressed on the surface of the cancer stem cells, and they're responsible to throughout all the drugs which they are being treated, and thus they become resistant to therapy. And these are certain signaling pathways which are available, which are present. These signaling pathways are majorly responsible to keep these cancer stem cells, to sustain these cancer stem cells. So each and every pathway is very important for the survival and sustenance and maintenance of cancer stem cells, starting from Wnt, NF-kappa beta, Notch, SHH, hyposignaling, and PLR pathway. All these factors together upregulate the pluripotent genes, which are NANOX, SOX2, OX4, KLF4, which are known as the heartbeat for survival of cancer stem cells. So if we can target these pathways, but it is very difficult to target all the pathways in one shot. Till date, we have no therapy which can target all these pathways in a single shot. Thus, it becomes very difficult to target cancer stem cells from root. So, till date, we have immunotherapies, cell therapies, small molecule inhibitors, which are available in the market, which are there, which claims that they can target cancer stem cells. But unfortunately, what happens to a particular patient, you cannot give all these drugs together, right? So depending on the stage of the cancer, type of the cancer, a patient is prescribed 2 to 3 drugs, or maybe hardly a group of drugs which is specific for that particular patient, but those drugs will might go and target a single pathway of cancer stem cells, but all of the pathways together cannot be targeted. Thus, cancer stem cells escape anyway. So before ending my presentation, I would like to highlight that we at Exsure, we are developing a unique exosome-based therapy pathway which can take these chemotherapy drugs or any kind of small molecule inhibitors, anything you can pack within the exosomes, which I will share in my later presentations, that we at Exsure, we are developing a platform which is cell-free, a cell-free therapy which will go and target these cancer stem cells. And keeping my fingers crossed, we have obtained amazing results, encouraging results in mice model also. And we are looking to go to really take this platform to the clinical trial level. So that's all from my end. I would like to thank everyone for joining. Thank you all for being patient.